Okay, guys, part two of our meiosis screencast. So we'll start with meiosis one. Again, this happens in two parts, meiosis one, meiosis two. The point of meiosis one is to separate the homologs or the homologous chromosomes. Now, this process starts with interphase. We talked about interphase in mitosis. It's where a cell spends about 90% of its life. More importantly, this is the phase where DNA is copied. So, interphase in meiosis happens one time. There is no interphase one, interphase two. It's just interphase, happens once before prophase one. DNA is copied, which will give the cell two identical sets of chromosomes. Okay, and we already talked about this. Again, this is the production of gametes or sex cells. This happens in oocytes, specialized cells in the ovaries of females, and germ cells, which are specialized cells in the testes of males. Okay, so let's go through meiosis one. We'll start with prophase one. There are some similarities here between prophase one and prophase of mitosis, specifically that the DNA will condense into the actual chromosomes, those X-shaped structures that we kind of have come to think of as chromosomes. They are paired with their sister chromatids. So again, sister chromatids are identical chromosomes. Homologous chromosomes are coding for the same things, but one's from mom, one's from dad. Now, what's different here from mitosis is your homologous pairs exchange DNA in a process called crossing over. The bottom parts or the lower arms of the chromosomes literally get tangled up, and when they come apart, they have exchanged pieces of DNA. Now these are called recombinant chromatids. These are no longer sisters, right? Because they're not identical. These are no longer sisters. They are all recombined. This is a way to encourage genetic variety in the sex cell. So this is the reason why, somebody in a family will look just like their great aunt or their grandma, even though the parents don't. This is a way of mixing up all of those genes and getting lots of variation. What they'll do at this point is form something called a tetrad. So these homologs, we'll just keep calling them homologs, actually join up so that they are adjacent to corresponding genes on the other chromosomes. A couple of other important things that happen and things that are similar to mitosis. The nuclear membrane comes apart so the chromosomes can be free. The spindle fibers start to move across the cell. So when it's time for those chromosomes to be pulled apart, the spindle fibers will be able to pull them apart. This is just another example of prophase one crossing over, which you can also refer to as genetic recombination. Next is metaphase one. The chromosomes line up randomly along the middle of the cell, meaning not all of mom's chromosomes are on one side and not all of dad's chromosomes are on the other. They line up randomly. There is no order here. And again, that's another way to encourage genetic variability. Spindle fibers attach from each pole to the homolog. So again, you can see those spindle fibers starting to attach so that they can pull those chromosomes apart. Anaphase, just like mitosis, A for anaphase, A for away. So here is anaphase one. So you can see, right, those homologs, even though they're not technically homologs anymore, the homologs get pulled apart. Each chromosome at this point is two chromatids attached by a centromere. So this is different from mitosis, right? Because the X's are getting pulled in one piece instead of being pulled apart. And again, this is random separation. So it's not all mom and not all dad on one side. They are random. This is something called independent assortment. And again, this is another way to encourage variation. There's a short video for you to watch that will explain this a little bit more. Telophase 1, the chromosomes reach opposite poles, cytokinesis begins. So, very similar to mitosis. The difference here, and this is a big difference, these new cells are going to be haploid. So, you see here, this cell had 1, 2, 3, 4, 5, 6 chromosomes. Each of those X's is a chromosome. Your new cells have 1, 2, 3. So this is an example of reductive division because you're reducing the number of chromosomes to half. Okay, moving along to meiosis 2. Meiosis 2 is a little bit simpler to explain. There's not as much going on here because kind of the big stuff has been done. The crossing over has been done, the genetic recombination, random assortment has been done. So prophase 2, spindle fibers form, begin to move chromosomes towards the midline. Remember, and I didn't mention this again, but I will, there is no interphase 2. You go right from telophase cytokinesis 1 to prophase 2. All right, so spindle fibers attach and start lining up. Metaphase 2, chromatids are lined up in the middle, facing opposite poles. Anaphase 2, A for anaphase, A for away. The chromatids separate and move towards opposite poles of the cell. So you can see here these X's are getting split up in anaphase. And then lastly, telophase 2 and cytokinesis 2, you get a new nuclear membrane around the chromosomes in each new cell. And what you end up with are 4 new cells, unique to one another and unique from the cell that they started from, and haploid. Each cell will contain half the original cell's number of chromosomes. So think about it like this, right? 23andMe. Humans have 46 chromosomes. All of our gametes, our sperm cells and our egg cells, contain 23 chromosomes. Okay, and the last thing to mention, and this is kind of a nice explanation of the whole process. You go from 46 single chromosomes to 46 sister chromatids, so that DNA has been replicated. Meiosis 1, you get 2 cells with 23 sister chromatids. Each of those split into a cell with 23 single chromatids. Now, the difference between males and females, other than the type of cell that is produced, for each cell in a male, you will get 4 sperm cells. For each cell in a female, you will get 3 what we call polar bodies and 1 egg cell. So this process will really, in a female, only yield 1 egg. The other 3 just kind of go away. The body will pick the strongest cell, the best cell, and have that one develop into an egg. And if you think about it, it makes sense because if a woman released 4 eggs every month, that would be a potential for 4 babies every time, and that's a lot of babies. So if you think about it from an evolution standpoint, a few 100,000 years ago, you don't want 4 babies, you want 1 baby. That's enough. Okay, guys, I hope this makes sense. We will meet this week and chat. I have lots of videos for you guys and supporting stuff. Let me know if you have questions. Bye.